So, good morning and welcome to this session on cell-based immunotherapies. I don't know whether you see the, okay, you see the slides. So the aim of this session, which will be shared not only by me, but also by Antonia, and we will introduce ourselves soon. So the aim of the session is really to explore ideas and directions for making the Human Cell Atlas more relevant, immediately relevant for the development of cell-based immunotherapies. This, of course, will have a focus on oncology, but not just oncology, also other diseases, of course, can be discussed. So maybe a very brief introduction, because we are both new to the Human Cell Atlas. So my name is Diether Lambrechts. I'm more an early adapter of single-cell work in solid tumors. And then we have also Antonia Müller, who is a MD and who has experience in stem cell transplantation and immune cell therapies, both in humans and mice. Okay, and now I will give the word to Antonia, who will give a brief introduction.
So I consider myself a physician scientist, but I spent the majority of the last 2 years in the hospital. So I will try to give you a really brief background on my idea of cell-based immunotherapy. The one big topic is an allergenic hematopoietic stem cell transplantation, which is not a pure stem cell transplantation, but if we transplant those cells after a preparatory conditioning of a patient with a leukemia, we give a cell mixture which comprises stem cells, but the stem cells in the graft are actually only like 1 to 3%. And then there is a whole mixture of other cells which we ignore. We talk about stem cell transplant, but it's mature lymphocytes, it's all types of other cells which we give in. And actually, to make this therapy effective, we do need mature lymphocytes, because mature lymphocytes, in the clinical scenario, they are mostly HLA matched. They need to be able to recognize minor antigens on leukemia cells to exert kind of graft versus leukemia effects. This is what will keep control or can control over leukemia after the conditioning regimen is over the months, and a single cell leukemia cell is hiding somewhere, and if it pops up, then the donor immune system can kick in and recognize tumor antigens or minor antigens, we don't know, and if we are lucky, then it'll take control over it. But there will be also mature lymphocytes which will recognize surface structures on healthy tissues and cells, and that causes graft versus host disease. And graft versus host disease can be really detrimental. I'm just showing you 2 examples of my patients from my clinic. One is like where you can see the cornea has several ulcers, is perforated, and with an eye like this, obviously, you can no longer see. And or skin here, the skin is so tight that basically they can't move. The side effects and the toxicity are the reason why we cannot apply an allotransplantation to more patients with, for example, autoimmune diseases or other metabolic conditions, which in theory we could probably also cure with a stem cell transplant. But this is not possible because of the toxicity. So if we think we do this because we want to cure leukemias, there are certain leukemia subtypes. If we treat them with chemotherapy alone, the cure rate is below 5%. If we treat them with an allotransplantation, with this immune therapy, then the risk of survival and cure goes up 20 to 30%. There's the ones who have a little better chances. But overall, we can cure approximately 50% of the patients with an allotransplant, meaning the other 50% we lose either due to toxicities and side effects or due to relapse disease. And we've become better over the past decades with handling infections and overall mortality, but we have not become better with controlling relapse. So my perspective and my view is we just put in cells which comprise stem cells that will be responsible for blood formation, and all the rest we have no idea. And sometimes it works, often enough it works, that's why we are allowed to do it, and often enough it doesn't work. So dirty in, dirty out. So one new form of therapy that is trying to avoid the toxic part of the stem cell transplant, but just using the immune therapy part, is chimeric antigen receptor T cells, which you have all heard were for clinically applicable or available CAR Ts. One takes autologous T cells from the patient. T cells from a patient who has seen chemotherapy, who has a disease, who has whatever, agents and drugs, we don't care. Just the number of T cells should be above 1 × 10⁹. Then that's okay. So we give in a T cell, and then there will be retro or lentiviral transformation or transduction, and then a certain percentage, like quality release is okay if the transfection rate is above 3% and shouldn't be higher than 25%, then they are released and given back to the patient, and then can target the tumor. And this is exciting, and it helps to treat a lot of patients who otherwise would have had no hope of cure. But again, I'm showing you the big 3 pivotal studies that resulted in their approval. You can see best overall response is somewhere between, let's say, 50 and 80%, but a durable complete remission is somewhere around 30 to 50%. Also there, we put in a CAR T cell, and we don't discuss any longer what we put in, and then we see what happens. And if we are lucky, it works, and often enough it doesn't. Then, of course, so we have the part where we should look at those adoptively transferred cells. What are those? Be a little more specific, learn a little bit more about that. And then, of course, how they are modified by the environment. Because, of course, immune cells are not a fixed cell, but their behavior can change over time and can be very plastic, or they can become activated, they can become tolerant, they can become allergic, they can become exhausted, and so on. So, and that was kind of the background introduction. If we start thinking about how we can improve cell-based immunotherapies, we thought, like, I don't know if you want to continue. I'll pass on.
Yeah, we can do this together. So, yeah, we identified there for a few key questions, but yeah, these are not the only questions, of course, but let me just briefly run through the questions and then ask your feedback. So one of the questions is how we can identify the heterogeneity in the cells that are transplanted, and whether this heterogeneity is, of course, also leading to variability in therapy response. And so the question here is whether the Human Cell Atlas, having annotated, of course, all these cell types, immune cell types, could contribute to this. I think that is an obvious question, of course. I don't know whether we first discuss the questions one by one. Maybe if anybody has feedback on this first heterogeneity question, we can, of course, discuss.
Help again from you guys, because I think talking for the clinicians, as a physician scientist, to me it's clear we have to go back in the lab now. We have something and we have a proof of concept, but we need to improve it, because just think, an allotransplant is about 300,000 euros, and a CAR T cell therapy, the product alone commercially available, is something between 350 and 400,000. So, and also, of course, those patients have really high hopes when they are treated, and so I think proof of concept is fantastic, but now we have to go back to the lab and do more homework to improve it.
We have one comment. Yeah.
Cells outside the body, you modify them outside the body, and then you put the whole mix back in, or you might FACS purify those cells that you feel are the most useful ones. But you could obviously also deliver the CAR directly as an in vivo gene therapy, in which case the whole question of heterogeneity is a very different one, because then it becomes like the moment the cells are outside the body, they're kind of a product that you can single cell sequence, that you can quality control, etc., and you put them back in. The moment that you do the in vivo editing, the whole question of heterogeneity becomes a question of gene therapy target specificity. And obviously then the
Not only target specificity, right? I mean, the target is defined by the
No, I
CAR.
mean the specificity of the lentivirus infecting its target cells, for example, or the gene therapy.
Yes, and the
delivery.
T cell features, because a T cell is also not a firm and stable thing, right? A T cell has seen chemotherapy before, comes from an old person, comes from a young person, and then just think of all the subtypes, subsets, the memory ones, the central memory ones, the naive ones, and there's many more markers now, like which, yeah, of phenotypic descriptions, but then obviously also functional descriptions. And so I think at this point we don't even know yet. We have ideas or hypotheses which population is the one that would work best once it's been genetically modified and put back. Yeah.
I think this is a great discussion. I guess maybe I would just say there's 3 types of heterogeneity that I can imagine, whether it's ex vivo or in vivo. There's the heterogeneity, and you've kind of alluded to all of them, but I think it's maybe helpful to just put them into buckets. There's the pre-existing heterogeneity of whatever cell population is potentially transduced, whether that's in vivo or ex vivo, all the existing states of cells that might be modified. Then there's the product or the resulting after the transduction or CRISPR modification or whatever it's going to be. So you have the pre and the post. And then there's the third, the sort of long-term heterogeneity of in the environment, the tumor microenvironment, the lymph nodes, wherever you need a site of action.
The tissue,
And I think all of those need to be quantified. And I think we still don't know. I mean, heterogeneity in itself might not be bad, right? You might need a heterogeneous collection of cells to be productive. But so that's where single cell measurements become extremely important for all 3 of these, to be able to define what are the right states that we want at each of those parts of the manufacturing process. And I think it fits, and I think another question will be, how do those states compare to states that are seen in other conditions, right? Are there natural states that in the immune system that we're trying to recapitulate? Are there new synthetic states that we want to push those populations to? Or third, like, are there even things that might be pathogenic in some contexts? So something I'm really interested in. Can we learn from autoimmune disease? You know, states that might be exactly what we want to engineer in a controlled setting into treatments for cancer or infectious disease. So I don't know, that's sort of my framework for this, but I'd love to hear others' thoughts.
Sorry, Andrew Farmer from Takara Bio. I'm not really in this field, so this might be just done, but sort of following on Alex's point about heterogeneity, I just wonder about, we're putting in a specific cell, this CAR T.
In the case of CAR Ts, right? In the allo transplant, we put in cells.
Right, but I guess my point is, I mean, we know that the immune system depends on multiple cell types to actually cause function.
Right.
So you may not be getting the function you sort of putting in a single cell, like a CAR T, may not really be the answer. You may want to be putting in multiple cell types with factors in them to actually really. So back to this sort of point of heterogeneity may actually be good.
The special recipe, leave out the ones that are inhibitory and put in the ones that are
need that balance.
helping.
I mean, I think we've learned a lot in the last 2 years from COVID and all the vaccine studies, right? I mean, those responses in people can really depend on multiple factors and different cells there. So I think that's something you have to, so heterogeneity is not bad, could be good.
Right, but the right one. And then,
Right
of
one,
course,
yeah.
the milieu also matters.
Right.
Yeah.
Hi, I'm Amanda Oliver from the Wellcome Sanger Institute. And I'm now working on kind of healthy lung stuff, but I did my PhD in cancer immunotherapy, and part of this, cell types, it's also, if we're engineering cells, we can engineer them to orchestrate the milieu or to, get the right cells there and polarize, manipulate that.
We have drugs. We have drugs that we can use to polarize and
Yeah, but not just
influence
drugs,
the
like also
milieu.
genetic engineering.
Yeah,
We
right.
engineered the car, and this is a whole field
Yeah.
of research in itself with multiple facets.
absolutely.
But cytokine engineering in there, all these kinds of things. John.
Thanks. John Bashkin with S2 Genomics. I have a couple of questions. One is, are you reliably characterizing the cell type representation that you're giving back to the patient?
So the thing is, we don't, and in fact, if we order it from some company, we get some information back. From the other one, we just get within label and zero information back.
Okay, so...
And then technically, we are not even allowed to take a bit of the product and analyze it, because it all needs to be given back to the patient. So,
Right, so during the initial isolation of the TILs or the immune cells from the patient, that's going to introduce a representation bias from what the patient originally has. And then I'm wondering also, because when you do those isolations, you can also induce a lot of cell stress just through the isolation process itself. And I'm wondering how much memory there might be in the immune cells from that stress response that is something you need to try to avoid.
Right. Of course, and then when you transfuse them back in, at least in mice, you often see that they convert over to memory phenotypes right away. Those are all things where I guess we all here agree that those things are happening. And I think we have a lot of technologies and tools to influence them now. But before we can influence them, we need to understand what's going on.
And you have to have control over the process so that you can then systematically manipulate the process.
Right, exactly. But this is big efforts, right? And
So it seems that in, 5 years, 10 years, we will have something that's like a fax machine, but with single cell sequencing, such that you can really, or like a flow cytometer, I should rather say, not sorting compatible, but reading compatible, that would just read 500 genes or 1,000 genes, etc., based on their kind of mixture of protein, RNA, and give you 50 million cells counted, basically. Once such a machine exists, what would be the amount of evidence, or what would motivate a practicing clinician in day-to-day practice to characterize the blood sample? Let's just leave the CAR T cells out because they have this commercial dimension. But in a bone marrow transplant setting, what would kind of convince you that it's worthwhile spending 1,000 euros characterizing the bone marrow if with such a technology where you know precisely what you're putting in, or would you say you're so happy that you found a donor, this is the last resort anyway, so you put the whole thing in, and anything that would give you further characterization would just give you a headache, but you're not quite interested.
No, so I think that is what makes being a physician scientist so interesting, because I would give you, if you had the machine, I would give you every single sample, because I'm also the one who sits there then after 6 months saying, okay, you went through hell, but the disease is back, and now, you can take a vacation now and spend your time, and you are the ones who have family sitting there, and all of a sudden, before you were very strict, and all of a sudden you explain them how they die. So those are the moments where I feel like I'm glad that I have a research background, because I understand a bit of the biology behind it, and I believe that maybe I can contribute a little bit to improve things. But very clearly in the clinic, we feel it every day. We need to improve because we tell them when the therapy failed.
But isn't it possible to sort out the bad heterogeneity, so to say, and then to implant?
If you knew. Technically, there is high-speed cell sorters for clinical application. They are available. But for that, of course, you would know which populations, and then, of course, you would need a lot of money for a clinical trial because all reagents you use need to be clinical grade, need to be EMA approved, and so on and so on. But I still think that this is what should be done.
Yeah.
And it cannot be done everywhere at the same time, but technically, those high-speed cell sorters have been around for 30 years.
So we're in this strange phase at the moment. I think of medicine as either procedural or product-oriented. Procedural is surgery. You go to the master of the field, you learn how to do a certain surgery, you move on, and the surgery is the way to do it is in your brain as a trained scientist. Product-oriented is something that's run through the FDA, and afterwards anyone can give it. Obviously, still, but primary care physician, local oncologists can use pretty complex therapies that they often don't understand what they're actually doing, but they're still allowed to treat patients with that because the idea is that the FDA has done the checking on it. Now bone marrow transplant was very clearly within the realm of procedural medicine. And now over the last year, 10, 15 years, we've seen a creep towards product-oriented medicine. And you said the extreme case that you're not even allowed to check on the product that's being used.
For the stem cell transplant, yes, not for the CAR T. For the CAR T,
for the CAR T cell. So in a way, a lot of academic expertise is taking out of this equation. So
not
what's
making us
the way
happy at all.
forward?
Yeah.
Is this really unavoidable and the right thing to do, or how would we make sure that all the information that is being generated in the Human Cell Atlas, in these type of initiatives, would that always have to go through the funnel of commercial development and then 10 years later come back, or there's a way how we can keep this procedural mindset to cell-based therapy?
I mean, we cannot analyze the product itself, but once it's infused, we have access again to patient material, right? And of course, the cells, before we send them off, we can also analyze samples. And then, of course, I think it is, the pharma companies had the big advantage in this field because they are super fast and they have money. They invested a lot of money and they haven't made any profit out of it yet, but they are super fast. We saw this with the vaccines and regulatory people and so on. We are slow. This is why they dominated the field. I think now academic institutions start thinking if the applicability will be much, much bigger. It will be so budget relevant. At some point, we have to think about academic production. And that, again, scares pharma companies. And also at the beginning, it was one company. By now, it's 4 companies with products out there. And if you look globally, it's even 5 or 6 companies. So I think now they have to be a little less dominant and have to agree on our demands and wishes, where we say we want access to our data, we want to know what we infuse, and we need to know. So I think that over time they will be, some companies are happy to share the data and others just say, no, forget it. But I hope that they will change too, and will have to change.
I like this framework about thinking about the balance between procedure and product. I think it's a really useful way to think about it. And I think in some ways the aspiration is that to have a product. If the product were extremely effective, it'd be nice to have something that were defined and you could just give and count on it working and have a minimal release criteria to make these maximally accessible and easy to deliver. We are in an in-between place right now where we're giving things that are products, even the commercially available products are given as a product, but they're still heterogeneous. If it were a pill that had this level of heterogeneity, it would be a very unusual situation, right? When we take a Tylenol, we expect it to be consistently a Tylenol, but that's clearly not the case for CAR T cells that are autologously manufactured. So I think that we need to keep both goals. One is to try to figure out processes that you alluded to that would give us more defined products so that we can have more reliable outcomes and that we can expect and that it'll be better, but also have the nimbleness for procedures. And I think I'll stop here, but I think what distinguishes this from small molecules is that the types of procedures can be quite rational. Now with gene editing and sequencing, we kind of start to have a huge amount of knowledge about how we can start engineering cell states. And so I think that we want to have the maximal flexibility from the full academic community and industry thinking about how do we shape these products going forward and continue to be. So I think getting the information about every product continues to be an iterative process of which are the products that are effective and how do we keep engineering to that. And then hopefully we lock in products that really work safely and effectively.
Right, and I think if I can just comment back on this, I think CAR T cells are here to stay, and I think once we solve the problem for solid tumors, then they will really expand, and I think for some diseases they have the potential to finally replace ugly chemotherapy, and that'd be such a blessing for cancer therapy that I think it's worthwhile figuring it out and solving the problems. And I also think that this needs a let's say global or international effort, because single institution efforts will just not lead to the solution in a timely manner. That is why I think things like the Human Cell Atlas and ideas like this, who really want to obtain as many data as possible, or
I think some mix of coordination, but also nimbleness that we're making sure that we're
of
trying a huge
course.
Yeah.
But
Some of this can be done in diverse settings with lots of ideas being tested.
yeah.
Yeah, go ahead.
My name is Jun Seita from Riken, Japan, and the one thing I'd like to make a comment is the Human Cell Atlas project is about the healthy human cell atlas. So I think that now it's, and it looks that the Human Cell Atlas project itself is now getting mature, so now we have to start thinking about the disease cell atlas or something like that for the next effort. And I'd like to know the current situation in the clinic. So when you choose, for example, the CAR T therapy to this patient A, so what is the resolution or the information that you know about the diversity of target cancer or the target disease? I know that there is the criteria.
So of course there is the label for which indications this specific CAR T has been approved. Doesn't necessarily mean that all CAR T cells are approved for CD19 positive malignancies. So, but it's the pivotal studies, and if they included DLBCL, diffuse large B cell, but not primary mediastinal, then you kind of have to stick to the label, or you have to convince the insurance company to still pay for it. And then the line of therapies, how many prior lines of therapies did they need to receive before? They must have seen rituximab, they must have seen anthracyclines, and so you have a list of things that they have to fulfill. But this is not scientifically based. It's not that you have a biological evaluation where you say, okay, we look at the efficacy of the T cells or their potency, and because they've seen so much bendamustine already, they are already so the probability that they will be successful and sustain a long-term immune response is so little, because we don't. So the selection criteria are purely clinical and have nothing to do with biology.
So how difficult to get some of the cancer sample from the patient and apply, for example, the single-cell sequence?
Well, usually you have the sample because you have usually at least the big university centers or the big centers want to confirm relapse and take samples if the lymphoma is accessible. And usually if you are at a major center, I would say that most centers try to snap freeze also fresh samples. I guess
Yeah, I think so.
And it may not be in smaller hospitals, it may not be in private practices, but I think the big centers aim for that.
You might need some coordination to get high patient numbers overall,
Yeah.
but I think it's definitely feasible.
Right, so, and I think all the experts in big centers consider this standard of care, which should be done whenever possible.
Yeah.
There was a question in
yeah,
the
there
back
in the
for a
back.
long
I
time.
will-
So,
Hey, I'm David from Hematopoietic. I was wondering, because I guess you two have probably the best intuition for how the cell system works. If you had to take a bet, would you say that given advancements in heterogeneity characterization, is there more potential in changing this in the dish or to influence the sort of like evolution of the system in the body with like, because I guess you could influence the way the product develops in the body with like
So
small molecules.
I would put my money on in vivo modulation. I don't know how you see it, but I would go with the in vivo modulation.
Yeah.
Because tuning something in a dish to get the release criteria fulfilled, I think that is relatively easy, but what you need is, I think, the impact of the environment and long-term sustained immunity is more important.
I think it comes down to selecting the right immune states from the patient, no? That seems quite
probably,
obvious. Yeah, I mean, there are several other comments.
Maybe a little, I know you ultimately want to cure cancer and focus on that, but I mean, something that interested me recently in the CAR T space is people using CAR T for other things. So there was a paper in Science for treating like cardiac fibrosis. I just thought that was really, just broadening out CAR T, but cancers itself is very heterogeneous, right? If you're looking to get broader sample sizes and something more, say, I guess, tractable to understand, maybe some of the learnings from successes in treating things like that might help us to understand what we should do with CAR T for cancer.
I mean, I'm interested in cancer because I'm a hemato-oncologist, but of course, CAR T cells could be applicable also for certain infections, for autoimmune diseases and so on, which again would be a very good thing, increase the number immensely, and therefore we need to streamline the processes, make it much, much cheaper. I think from a medical perspective, they have a super broad application or potential for applications and should be pursued, but yeah, absolutely.
If there's not too many side effects, I think that's also
Right,
important.
yeah, that's a topic we haven't discussed yet, the off-target or on-target and off-target effects.
Yeah. Okay. There's still one comment there, and then.
So, sorry, this may be a very naive question, but have people, presumably people are doing single-cell sequencing before and after treatment. And so if you're seeing changes that are long-term in open chromatin regions of the effects like that, are people seeing that? Are they doing that? Again, thinking of how does the HCA contribute to these efforts? That infrastructure would seem to be an appropriate way to connect.
Yeah, I think this is exactly kind of the thing we were thinking about. So you characterize at single cell level the cells that you deliver to the patient. You follow them during treatment by taking these on-treatment samples. You characterize the states, see how they evolve, and from that you can learn the right heterogeneity that you need to deliver to the patients, and you can do this
And I'm not sure, I haven't seen data where people do it. I guess the chance that people do it is probably bigger for CAR Ts because CAR Ts are more sexy than an allotransplant. Allotransplant, I'm not aware of data, and the problem there is that a kind of clinical transplanters feel they are already pretty scientific, but we have zero knowledge and skills dealing with big data and doing all these things, and that is where an allotransplant are no longer really sexy, but CAR T cells are only there for very distinct diseases right now, so we will not replace allotransplant in the next 10 or 20 years. I'm pretty certain about that.
Hey, I'm Wen from SAM.
Yeah.
So far we are talking more basically about the autologous, right? T-cell therapy. Autologous.
The CAR Ts, those are the commercially available ones.
Yeah.
Allogenic ones
Yep.
would be much cheaper, and that's. But there's some medical hurdles to that, so it'll take a few years until they make it in the clinic. Yeah.
Okay, I'm thinking if like which HCA can contribute in this side, could it be possible like we just to gather the off-the-shelf product and based on that to study which the case is most benefit in the clinical trial, and based on this, so we are talking about this part about heterogeneous. In this way, it's already we focus on one potentially product in the future, and based on this to improve, it more makes sense to rather than collect the sample, let's say the sample from the autologous case.
Yeah.
Does that make sense?
Yes, I think
reduce
I think I understand what you mean. And I would say probably let's not separate allotransplant and CAR T cells that much, because I think we can learn a lot from each other. The biggest hurdle to allogeneic CAR T cells, third-party allogeneic CAR T cells, at this point in time is that they will be rejected probably. That and overcoming the genetic barrier to get them engrafted not only for a short period of time, but for a longer time periods, probably months to years, is a big hurdle. And then, of course, the safety issue, that you have to be totally sure that you do not give so if you have a manipulated cell, then probably the CAR is stronger than the endogenous T-cell receptor. But in your product itself, in the currently available ones, release criteria are that the transduction rate should be between 3% and 25%. So there is a lot of unmanipulated cells in there. And even like very, very few normal allogeneic T-cells, non-HLA matched, can be detrimental and deathly, and so those are big hurdles. So if we improve the autologous ones so much and go into earlier lines of treatment where we don't have to have given a lot of chemotherapy before, or we collect it upfront, then maybe and we get rid of those long production times, like 10 days in vitro, another 14 days quality release, and so on. And they are developing platforms where you really do most of it in vivo, the in vivo expansion. Then the production process is like 2 to 4 days, and then quality release, early release can be after 5 days. So I'm not totally convinced yet that the money is in the third-party allo CAR Ts.
Yeah.
Thank you. Yeah. I'm Artem from Boehringer Ingelheim here in Vienna. So a bit more of a technical question about the heterogeneity. So if we are interested in, if there is a heterogeneity in what's being injected, would it make sense for the manufacturers of the cell therapies to do some sort of barcoding of what's being injected? So if something goes wrong, we can basically go back and see and track what's in the original, mix was, if there is a specific clone or something
Absolutely.
Like that, that's triggering the.
But at this point, they want to sell their product because they are pharma companies.
But do you think they actually do that? So, I mean, it is possible that there is some barcoding thing, and they just
Can you
don't
do it
tell
in
where
humans?
it is.
Can you give back a barcoded product?
In humans?
You can give
The
it back
question
in
is,
humans, right?
I was also
Approved,
wondering
no.
if it's like, if there is a safety risk here, or like theoretically it's not that.
Well, but you have to go through the approval process, right?
FDA
It
approved
needs to be GMP, clinical grade.
Yeah, that's a good.
But I mean, those are exactly efforts that this is super interesting ideas. I mean, and I think
This
we
is what-
should think about this type of ideas, like how could we
This is what needs
follow
to be
things
done,
and
but
learn.
yeah, it's over there. Yeah.
I mean, there is work on this, right? So the other form of natural barcoding in the CAR T cells is the TCR repertoire. And Penn has been a big leader in looking at the TCR repertoires that expand during CAR T cell treatment.
Yeah.
And that's led to these identification of specific subclones that grow out. And actually, one of the reasons for that has been sites of random integration. And so notably disruption of the TET gene, for example, was picked out because there was this winning TCR, which was just essentially serving likely as a barcode for this integration site. So you can either go either way, use the integration site as a barcode or the TCR. So I think that there are more opportunities, especially in academic settings, to think about how do we be nimble about thinking about a number of different technologies that we've heard about natural barcoding and other types of things to try to be creative. One of my big aspirations, which will be a big regulatory hurdle, is can we actually start designing mixed products and see which ones win out in patients so that to accelerate the clinical development time. So, if we don't know up front which types of products will be best, can we put a mix in? And either will that heterogeneity of product be useful, or can we just see that a subset of it was the part that actually wins out in vivo? So I think there's lots of opportunities for HCA to be measuring these things pre and post, to be learning about what's actually effective.
Yeah, no, I agree. Yeah, I will guide it back.
Graham Heimberg from Genentech. I guess I was kind of curious about, let's assume we can characterize the heterogeneity very well, and we see all these diverse things going on. Is there any way to, I guess, tie it to kind of the outcome? And I guess I say that besides, I guess, putting it back into a human, because at least on, let's say, other diseases, I think of this kind of analogous to a biomarker study. And, biomarkers, we don't always find good biomarkers, and I would say this is probably more complex given that there's a tumor involved, and then also, people on the other side designing the therapy. So maybe I'd say like 3 times the complexity of a normal biomarker study. And, you can't necessarily test that out in enough humans.
I mean, the advantage here is that we have all the clinical data, because whoever gets a cell therapy needs to be put into a registry. So in Europe, it's the European Association of Blood and Marrow Transplantation that collects all the data, and the database and the information we have to put in is extensive. I mean, this is a lot of work for the data managers, but we all agreed we need to have a lot of data, including cell count, cell numbers before, and so on. So the clinical database exists, and this is actually a great opportunity. We would just need to have like projects and link it to that, but the data are there.
I mean, does that have sufficient data? I mean, the clinical metadata is helpful, but if you don't have, let's say, the characterization on that person, then...
Like what additional data? Date of birth, age, disease, prior chemotherapies, survival, relapse, and so on.
But you want some characterization of the heterogeneity of what was put into them.
Yeah, but that's
No,
kind of what
only
propose,
like
huh? Yeah.
T-cell counts and that type of stuff, like the rough data.
Yeah.
I mean, for many, enough, if you try to pull an effort, enough people have frozen samples. Because whenever we freeze a product, we have to also freeze pilot samples for quality control. And in some centers, like in my center where I was before, we stored them for forever, for 25 years and 30 years, we have them in the freezer. So I think if you asked around, you would, and of course, in the clinics, we're always super busy, but enough of us think that way and know that this is precious samples and we collect and have biobanks.
I guess I'm a little worried that that may not be sufficient. I mean, look at PD-1, for example. Our best biomarker is PD-1, and we say that, you know, we think about that because that is the target. But ironically, even that is not a great biomarker, and, you know, we have poured a lot of money, I don't say we as Genentech, I mean, including Genentech, but we as a whole world have poured a lot of time and money into that, and I'm not so sure a ton has come out of that.
That is where I think I agree, academic, single-center academic efforts are great, but I think it also needs like bigger approaches where we think systematically and think like what would really make sense, and then I think, I'm pretty sure enough people would say, I can't run the study, but I'm happy to send samples and participate.
I think Alex's mixed product idea, you know, would be a faster way, but that sounds, I don't know how you essentially propose to do pooled CRISPR screens in people, so.
So yeah, I mean, there's a group of us who are crazy enough that we're starting to push for this. We'll see if there's ways to do it, but I really like your idea, Graham, and I think that this is something that the Human Cell Atlas could do, right? Is really think about how do we systematically make this database of pre and post infusion and couple it with the clinical data, right? And so, you know, and ideally we would couple it with, whenever possible, some kind of biobank repository for lab-based testing of these products, because one problem is we still don't know what the best models are that will be most representative of what's actually happening in a patient. So if we could have a big database of infusion products, tumor samples or even blood samples, couple that with clinical efficacy, and then you take those actual products and you see how they perform in both in vitro and xenotransplant mouse models, and really put that all together, I think we could start to learn lessons about which cell types, cell states are most predictive of positive outcomes for specific indications.
Yeah, yeah. It's kind of what we had in mind.
I mean, how much is the whole genome sequencing or whole transcriptome being done on these tumors in the, I mean, just, and that's why, when back to things like the fibrosis, and my ignorant thinking that that's a simpler thing to treat than the heterogeneity of a cancer, and that there's so many routes for the cancer to escape, that, and you're looking at ends of one in each patient you treat, right? So it may be very difficult to understand the reasons behind, you know, success or failure.
So I think right now, currently in clinical routine, it's probably rarely done. But I also think that a lot of centers come up with new programs for precision medicine and so on. So I think a lot of people think that way. So if we all actively promote it and somewhere, somehow also generate the funding resources for it, then I think you would probably have a lot of open ears who'd be willing. I think it's the practical thing and not the theoretical idea behind it that prevents it right now. So if the discussion slows down, should we look what
second
we have
one. No, I mean, maybe one thing we didn't discuss is that this heterogeneity can also be responsible for side effects. So, I mean, linking the heterogeneity with not only with treatment outcome, but with side effects that you have in the database, I think could also be very valuable, and identifying the states there that cause this. It's something along the same way, but I think that should be obviously part of that. Okay, you still had a comment?
So I think related to this question of side effects, this came up in the questions after my talk. I think we talked about one type of the heterogeneity product, but I think target design is another major place that the Human Cell Atlas could have a massive contribution.
Okay.
Is, thinking about how do we pick out targets, like if we're doing massive maps of expression, RNA, protein expression in tumors, for example, what are best distinguishing tumors, and where will that have predicted off targets? And can we find either individual expression of splice isoforms or specific expression of things that could be safely used, or do we need to find combinatorial codes and use an AND gate?
Absolutely.
And so I think that if the Human Cell Atlas could also provide an atlas of CAR therapy to essentially come up with a library
of the healthy tissues.
Exactly. Of how do we target to healthy tissues, or how do we target to any tumor? And having that kind of addressing code would be a very powerful analytic tool.
Because this topic here was a question in the back, or a comment. So that would broaden up immunotherapy or cell therapy to solid tumors, because most of those antigens are only weakly immunogenic and are not exclusive, and that's the major hurdle regarding safety issues.
I was wondering if it's understood whether the side effects are because of off-target effects of the cells that you're administering, attacking something they're not supposed to, or is it because of the host immune response?
So it's not off target, it is on target, but off tumor, right? Those are the key problem. The immunological side effects, I think, clinically we can handle them. We have some drugs, and we always have steroids, and there is now prophylactic and preemptive issues. But just to mention one published case report, published many years ago, one single case, that was a patient with metastasized colon cancer, HER2 positive, metastasized to liver and lungs. She got a HER2 CAR T cell, and after 5 minutes, she became short of breath. After 15 minutes, she needed to be intubated, and on day 5, she died. And then they did an autopsy, and they found that HER2 was also, like, at very, very low levels, expressed on endothelial cells in the lungs. So she went into vascular leakage, lung edema, and could not be resuscitated. And this is just published a long time ago, but this is what can happen. And before, nobody was aware that HER2 was expressed at such low levels on endothelial cells, and that's exactly the problem here, where we also need to know what is happening on healthy tissues. And the immune effect is so strong, so much stronger than a naked antibody.
There's also a few examples where the target is expressed on neurons, and where the patients then develop Parkinson's disease or something similar. And so I think that's where the Human Cell Atlas, I think, could really contribute, because you can monitor target expression in healthy tissue across the body, and kind of start developing new therapies with new targets in a much better way, I think.
Because, I mean, this is what you typically would need non-human primate studies for. And for example, I mean, I just moved here from Switzerland, and for a lot of things, they don't allow non-human primate studies, but they request them, and so they say, go to another country, do it there. But if you really, and also, of course, as a clinical scientist, you feel uncomfortable if you don't know the targets, and this is, I think, where the Human Cell Atlas could be really of help, if you kind of have a screening of all tissues before, where you can test it.
And I agree, this needs to include splicing and other non-coding RNA transcripts maybe that are often expressed. So I think it has to go broader than just gene expression and read counts, I think.
So does that imply then that the HCA should start doing other things besides 10x Illumina, you know, and start incorporating more
Yeah,
long-read sequencing.
It would require SmartSeq, yeah.
Yeah.
So maybe some SmartSeq, yeah, should be helpful. Would be helpful, I think. At least to address this question, because the splicing is difficult. I mean, you can detect some splice events with a 10x, but not for the whole gene. So.
Just a quick comment in the same vein of the splicing. I mean, we're talking about a healthy atlas, but often, you know, a cancer patient may be a 60-year-old person who comes in with, you know, maybe liver fibrosis or whatever. They may not even know about it. And so, you know, I think when you look at target identification in the healthy human cell atlas, you're going to run into kind of the same issues, and you may not even know kind of, they may be inexplicable at first because of the lack of knowledge of maybe a condition somebody has. So I wouldn't say, oh, we need, well, I mean, sure, we can do more in-depth splicing characterization, but that's kind of a drop in the bucket of all the kind of target characterization that, like, unknowns that you're up against.
And this is why I think it'd be desirable if the human cell atlas, once you guys are done with the healthy cells, also look at disease conditions.
On it.
There is one in the back. I know. Okay, I thought you were raising. No.
Do you want to take the next topic? Or I think we...
Yeah, so one other thing we discussed is kind of the immune suppressive mechanisms that are present in the host, more in the tissue. So there have been a lot of publications and reports that, for instance, resident macrophage populations can be either immune suppressive or the opposite. And I think characterizing the tissues to a better extent could actually, yeah, probably enable more tissue-specific delivery of some cellular therapies using enhancer-based developments and things like that. So I think that, yeah, much better understand what is present in the tissue, what is driving immune suppression, and these things probably could also contribute. And this is something where the human cell atlas would be perfectly positioned to do this. So characterizing the host tissue, comparing the different tissues within the healthy human body would be very relevant also, I think.
Because there are some organs, for example, the bone marrow, which is a pretty tolerant place, has more memory T cells, it has more regulatory T cells than, for example, the lymphoid tissues. And of course, just think of the placenta. That is like the major tolerant place. So it's not like an equal distribution of immunity throughout the body. And I think those things would be of importance and relevance too.
Yeah. Is there any feedback or comments on that? I don't know how far do we have.
So then we have more topics if the discussion slows down. But the discussion is already fantastic.
Has been great, yeah.
It's fun, yeah.
So we also identified a few potential solutions, but I think they more or less are along the same lines as what we have been discussing so far. So a potential solution or project, so to say, is to develop a curated database of immune cell types and states across tissues. And this could act as a reference for transfer learning when typing patient samples. So this goes back to the heterogeneity. So typing the heterogeneity, typing the immune cell types or states that could trigger side effects in patients. But then thirdly, also developing more the trajectories of the immune cell types. And this can basically reveal new states, the understanding of which may lead to improved cellular vehicles for anti-cancer vaccines or T-cell-based therapies. So I think this first solution is really based on annotation that you can do in the human cell atlas and which could really help and contribute to this problem. And then, a second potential solution goes back to the side effects, but this has also been discussed. So I've just highlighted here one paper that was published in Nature Medicine, where there was really side effects developing due to on-target effects, but on target where the target was expressed in this case in the brain, and where the patient developed, several patients actually developed side effects. So I think this was discussed, could also be a very helpful contribution of the Human Cell Atlas. And then we have a few smaller topics, but also these I think we discussed. So developing cell type specific enhancers or enhancer-based therapies could be very useful by studying these enhancers in the Human Cell Atlas. I think this could improve the development of these therapies. I think also identifying novel cancer antigens, this was also discussed, I think during the discussion, would be very helpful. So they would be upregulated in tumors, but absent or very lowly expressed in normal tissues. And this, of course, is not only targeting the coding genome, but also splice variants, long non-coding RNAs, etc. You can take this as far as we wish. But yeah, this was also discussed, so some smart-seq sequencing would be probably helpful there. I also think that on the healthy individuals that we study in the Human Cell Atlas, we should be doing extensive T-cell receptor profiling and HLA typing. I'm not sure this is included in all the studies.
And over age, I mean, in young people and old people and so on.
Yeah, so aged versus younger people. Again, because this is well established, so these tumor-specific TCRs are very frequent in tumors, but they should be absent or not clonal, at least, in healthy individuals. So also there, I think the Human Cell Atlas could maybe not drive, but be a helpful way to identifying TCRs that target tumor-specific antigens. I think this is all along the lines that we discussed. And then characterizing the tissues. So there is this emerging field of resident macrophages that can stimulate tumors or that can sometimes also be anti-tumorigenic. And so characterizing them across the different tissues in the Human Cell Atlas could actually be very helpful, because it can allow us to understand why in some patients there is not a long and sustained response to these cellular therapies, and this could be due to the host, and due to these cell types that can be pro-tumorigenic or anti-tumorigenic in these patients. And then I don't know whether I think this is more or less everything that we discussed. Did we miss anything, or do you have other comments, ideas? Yeah, there's one here.
Hi, I'm Eugenia, Center for Molecular Medicine here in Vienna. Just a question that I'm curious about. So this is a lot of exciting things that we want to learn about and can do partly in the academic setting, and perhaps even get some kind of HCA support first for many questions, and then extend to, I don't know, the Human Cell Therapy Atlas eventually with governmental support. But I'm sure also that the pharma companies would have the incentive to answer all of those questions. But is there any motivation for the pharma companies to make the data available? Because I'm quite sure they would do a lot of those things for themselves because they want to improve their therapies. The question is why wouldn't
You
they
know,
share?
in the commercial setting, they only can, they have material, they hold it back, but they cannot use it, right? For the studies, yes, because they have informed consent, but for commercial use, they are limited for regulatory issues. And because it's not a study, you cannot, just because you have cells, do with them what you want. It needs ethical approval, and that is a study, and in a commercial setting, you can't do it. And of course, they are kind of interested in it intellectually, but their priority is A, finally make some money with it, and B, design new cars and think of the next generation cars and so on. So exploiting clinical data to which they have also no access is not their priority.
But then this seems to be a very unfortunate side effect of this productization of CAR T cells, that it basically destroys kind of incremental learning,
Absolutely.
because you, the moment things on the market, the company does not want to change it, because that would be costly. They just want to sell it, which also means because they cannot change it in the short term, they don't even want to know where the problems are. Because the moment they knew where the problems were, perhaps fewer patients would be eligible and whatnot. So
I think they want to know, right? But they can't do it because it's not a study and they don't have access, and they are already in their brains and in their research and development, they are already like 2 lines further down.
But why did the FDA not ask them to do like a stage 4 study following up in a large, because these were such novel therapies, it would have been entirely reasonable to ask that very detailed follow-up is necessary.
They do this, so and then that is why there is the contracts that EBMT and CIBMTR in America do the clinical follow-up. So for each patient that we put in the registry, we get 2,000 euros from the pharma companies, so that we put the data in the in the registry, so that we can do post-authorization studies. But they only involve clinical data, and they do not allow the pharma company to do extensive studies on the cells that they have in the freezer. And of course, if you came up with a project for it, then they would probably financially support it, because they want to know. But if they have to put their money on something, then they want to earn some money with the product that they have and don't want to develop that further and want to have a new product that they can bring on market within the next 3 to 5 years.
Do we need policy change? Is this what I'm understanding?
Absolutely. Or we need to take it back into clinical academic hands, because I mean, we cell therapists, we can handle cells, and right now all the regulatory hurdles have been pushed so sky high, also a bit from the pharma companies, that academic centers can't do it. If we do a bone marrow transplant, a classical bone marrow transplant, we doctors, ordinary people, go in the OR, and then we have our drilling tool, 2 of us, and then we drill, and then we have a 20 milliliter syringe, and then we pull out bone marrow, and then we have this sac with a filter at the bottom. The sac is open, and then we plunge it in, and that is what we do. And then once we have like 1.5 liters, then we filter it, and then we infuse it. So and it works, and it worked for 50 years. And but if we asked for approval for this procedure now, it's a completely open process, just in an ordinary OR. We would never, ever, ever get it. We would have to go into GMP clean rooms, class B, and this and that, and this and that. And so the regulatory hurdles are so sky high that academic centers can't do it. Also because then it is, all right, you have the facility, you do it, do a study, do the first 15 or 30 patients, and then we evaluate whether we cover some of the costs via the health insurance. But there is no funding organ that will fund a clinical study with that. I mean, you need like millions, you need like 20 millions to run a study like this. And so the hurdles are immense.
So patents will run out at some point, and in the drug small molecule field we have generics, and generics drive down cost relatively quickly. What's going to be the generics business model for CAR T cells?
I don't think that'll exist, because the commercially available CAR Ts right now are like scientifically 12 years old. So nobody wants to rebuild those. We are interested in finding ways to speed up the incredibly fast research behind it and its translation into the clinic. Nobody wants to rebuild an outdated, very old CAR T, and all the new ones probably have patents again on it.
So are you saying, because in drug development, you have this chemical matter patent that really dominates everything, and when that expires, then it's free for generic. Are you saying, I guess one way to interpret what you were saying is, patents don't matter too much because you, with old technology, and you can basically work around them?
No, I
Or
want the
you're
new one.
saying it's a hopeless case to ever do generics because it would always be behind the state of the art?
Exactly. What I want from the regulatory side is that you do not, when you do a modification, only a modification, if, for example, the target is the same, but you modify the production process or minor changes that you don't have to go through the whole approval process. Like the flu vaccine, also it doesn't undergo phase 1, 2, 3 every year, but if you do a certain modification, there is fast track approval. And I want that for cell therapies too, because then it'll be cheaper. If you do minor modifications with the same target that you have fast track access, it'll make it cheaper and it'll ensure that we don't work with the scientifically completely outdated products in the clinic.
And that could be the trick to get that barcode into the thing, because if you are saying there kind of 20 bases of random code could be in there and they don't make it's just a kind of a spacer, then eventually it might, I think it's not totally out of question to eventually sneak a barcode into some of these products. Because in the end it's just random DNA, which at that point doesn't really make a difference. But obviously I think it will be, actually this is a chance for kind of small countries, perhaps even like Austria, where you can really sit down with regulators without them thinking that they will create a worldwide precedence case. Whereas the EU, etc., the EMA, the FDA, obviously they have to be super careful that they don't misstep because a lot of others orient themselves with them. On the other hand, it seems that the door's wide open for kind of starting to engage scientific dialogue, because it's clear that the current situation is not sustainable. At the same time, kind of, I think especially kind of your field has shown that they can very responsibly do bone marrow transplant and many other procedural ways of treating patients. So you will be trusted also, kind of, if you spin it the right way, that kind of this new generation can be delivered.
Yeah, and I think it's a mistake to consider those cell therapies a drug, because they are not standardized. My expectation to a drug is that it is completely standardized and that I have pharmacodynamics, pharmacokinetics, and all these studies done, and you can't do it for immune cells. You can neither standardize them completely, nor can you do all those things. To me, it's a living cell, a living immune cell, and should not be categorized as a drug. So if there is no more questions, I have a question back. So I'm not part of the Human Cell Atlas group. So from your perspective, after our discussion, which I really enjoyed, what's your thoughts? How can you help us cancer scientists and clinicians? How do we approach this? How do we keep talking? And what is ways, how do we get together? Practically, or do we meet again in a year, or because I think it's always fantastic opportunities if people who actually have different perspectives on things come together and talk and inspire each other. So I think we communicated our wish list to you now. What do we need to do to convince you?
Well, is it possible to expand your informed consent? When you have a patient,
Yeah, absolutely. Yeah.
extracting cells that are intended to go into therapy,
Absolutely.
and your consent to
Yeah.
include the research aspect
Yeah.
to it, then
Patients will always sign. if you explain them the importance of it, I mean, they totally understand that cancer therapy needs to be improved or whatever, not only cancer. And 99% of patients, if you take the time and explain them why it's important, will sign everything.
Yeah, that should be possible to do. Yeah. 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 And do it multi-institutional, I guess, to increase patient numbers. And
Yeah, probably like already, like I guess local biobanking is a very simple step, but aligning like sample type, time points, collection time points, and aligning those procedures, also certain procedures of freezing. I think those things are the basis for future studies, that you don't collect stuff randomly and then you look what you have in the freezer, because we all have tons of cells in the freezers, but usually not really coordinated and therefore often enough not helpful. So I think having like within different types of working groups, aligning those time points and sample types and providing guidelines for that would be helpful, because then whoever sticks to that can participate then in later studies.
Trying to scope the problem in terms of signal to noise. How many patients do you need in order to pull an effective biomarker signal out from all the variability from patient to patient? You know, if you were to do a pilot project with 10 patients and do SmartSeq 2 and fully characterize all their splice variants and really go, you know, as deeply as you can onto those 10 patients. Is that enough to do a proof of concept, or does that have to be 1,000 patients?
What the biostatisticians will need to tell us. I mean, the first pilot start would be like take 5 where it didn't work and 5 where it worked very well, and then take the 2 extremes and then see whether there is a clear different pattern. That'd be the easy thing, right? If you don't see that, then you would have to go higher up with numbers. But the pilot would always be take responders versus non-responders.
Well, you don't know raw priori, but if you do 10, statistically, 5 of them are going to work and 5 of them aren't. Right?
Yeah, but you could do that retrospectively. You can freeze the samples. So.
Yeah.
Yeah. Okay.
Yeah.
Okay. I don't know what time.
So now it's 5 to 12. So we are a little bit early, but I think if you guys are hungry and thirsty and...
12:05, 10 minutes. Okay.
All right, then.
Okay. Thanks a lot for your
so
contributions and for the nice discussion.
Yes, it was fun.
Yeah.